Hi, good afternoon, everyone. My name is Meenakshi Sarup, and today is the 21st of August 2024. Meenakshi Sarup, and I will be going through a very quick presentation about the roles of the pancreas and the liver in terms of the regulation of digestion. Okay, thank you very much. Now, in this slide, we have the cabbage, and the cabbage has a lot of starch, which when broken down into the monosaccharides, you have the glucose unit. So we're focusing on the pancreas, which not just targets starch or the saccharides, polysaccharides, it targets multiple nutrients, as you will see further down. So this is the pancreas here, and the pancreas is found right after the stomach. Right below the stomach is the pancreas, and the pancreas secretes, in its exocrine function, it secretes into the pancreatic duct. And the first thing that happens, the pancreas secretes, is a lot of bicarbonate solution. So sodium bicarbonate solution. And this bicarbonate secretion in the pancreas happens to food, to the chyme, as soon as it comes out of the stomach into the duodenum, or the first section of the small intestine. So food goes through the mouth, goes through the esophagus, into the stomach, and as it comes out as chyme into the duodenum, or the first section of the small intestines, the pancreas would secrete through the pancreatic duct a solution of bicarbonate. And this will raise the pH from the acidic pH of like around 1 or 2 due to the hydrochloric acid to a more alkaline pH, like around 7, 8. So it neutralizes the acid and raises the pH by a lot. And you can see that you have a lot of carbonic anhydrase being produced, which helps in the whole reaction with carbon dioxide and water and carbonic anhydrase, and the forming the hydrogen ions and the carboxylic ions, the hydrocarbonic ions, and the chloride. So there is an exchange happening here with chlorine and hydrogen carbonate, and there's a lot of movement of ions. You have active transport, you have the of sodium and potassium ions for the gradients, you have the transport of chlorine for hydrogen carbonate ions, you have carbon dioxide. There's a very huge amount of ionic changes happening within the pancreas that allows bicarbonate secretions to happen. And this pancreas also produces trypsin enzymes. We will see in the next slide, in the summary slide, you'll see a breakdown of all the enzymes that the pancreas produces. But the pancreas basically targets all food groups except for fats. Well, there is fats here and there, but the targets are more carbohydrates, and there's peptidases for the proteins. And we're going to talk about colipase as well. So you have the enzymes phospholipase, the carboxyl peptidase, the trypsin, the colipase. So it targets all food groups and all major nutrients. So let's go to the, and you can see, before we go to the other slide, you can see the pancreatic duct here. So we'll go to the other slide now. So within this slide, you can see a summary of the function of the pancreas. And of course, the pancreas has both the endocrine and the exocrine function. The endocrine has to do with insulin and glucagon and secreting that's within the blood. The exocrine has to be as a form of secretion within a duct or not within the blood. And that's the digestive enzymes and the sodium hydrogen carbonate, which is the bicarbonate, which helps to increase the pH. The bicarbonate secretions, and there's a main enzyme of carbonic anhydrase, which functions within that chemical reaction of breaking down carbon dioxide and water. neutralizes acid from the stomach, allowing for a basic pH, ideal for the pancreatic enzymes to finish digestion of carbohydrates, some fats, and as we saw, some proteins in the peptidase. Then you have colipase, which is a protein cofactor which displaces bile salts, allowing lipase to access fats within the bile salt coating. So in the other few slides, we'll talk about bile and talk about the functions of the liver, and we will talk about what bile does to fats, and we'll go back and reference colipase, colipase further down. Then you have the liver, and we're talking about fats, and that's why I have a picture of a donut. The liver has multiple functions, in which one of them is digestive, and the liver deals with a lot of breakdown of waste, breakdown of red blood cells. You have the bilirubin, the nutrients, the drugs. It's more like a detoxifier. This detoxifying unit is the liver. And the liver within bile. Now, bile is produced in the liver by the hepatocytes, and it is stored in the gallbladder for further processing whenever it is needed for digestion. And the liver focuses mostly on glucose and fat metabolism, protein synthesis, hormone synthesis, urea production, detoxification, storage. So the liver has many functions, and the one we're going to focus on is bile, and the production of bile, and the storage of bile, and the gallbladder. Now the gallbladder functions as a bile storage unit, and it keeps bile and secretes bile through the common bile duct, which is this really small duct, which empties into the duodenum here. So you see the gallbladder, the liver, the pancreas are all very closely packed together and very within the same area, and it all empties within the same area, the duodenum of the small intestine. And you have the hepatocytes here. Moving on to the next slide, you see the liver. Now the liver secretes bile, a non-enzymic solution which contains bile, salts, bile pigments, and cholesterol waste products. So it's not just focused on digestion, but it's just whatever waste products the body wants to get rid of through the feces. The bile salts act as detergents which emulsifies fat droplets. Now fat droplets are very hydrophobic and clump together. Fat droplets. Like you see oil on water, the droplets are clumped together, right? And the blood is like a very liquid medium, has a lot of water. So you want to get rid of whatever is within the blood, whatever waste products. The liver will help to do that. And within whatever it is digested, and in the chyme, which is very much liquid, you have bile acting as a detergent, which breaks down these large fat droplets into very small droplets, increasing the surface area for these lipases to come in and digest these fats. So with the increased surface area, lipases can work faster, and within the optimum pH of around 7 to 8 due to the bicarbonates, you have a quick and proper digestion of fats. And you can see, well, you can't see, but further down, bile that is within the liver, and further down within the gastrointestinal tract, the GI tract, the bile is recycled and reused again for more further digestion of fats, and it's stored back into the gallbladder. And a lot of the waste products are just re-excreted. And bile salts are amphipathic, so they're both having a hydrophobic and a hydrophilic region, making emulsification of fats very efficient and allowing lipases to digest the fats. So the bile salt surrounds the fat droplets and helps to keep it into smaller droplets as compared to the bigger droplets that there was before. And you can see a summary here. So this is the liver, the stomach, the small intestines here, and the pancreas. And they're all very closely linked. And the pancreas has acini cells which produces the pancreatic enzymes, digestive enzymes, and the liver has the hepatocytes. And the pancreas has pancreatic amylase, the lipases, the carboxylic peptidases, the trypsin, which targets all of the different nutrients, carbohydrates, the proteins, the fats. The liver has just bile, and it doesn't really break down any specific nutrient. It just increases the surface area by which the colipases could come in and break down the fats. And the release, so the pancreas releases through into the pancreatic ducts here, and of course that which is in the small intestine, duodenum, and the liver releases into the gallbladder, which is stored and then released in common bile duct. And you can see here that this is the common bile duct, and you have the pancreatic duct, which release basically in the same area. And they both have a pH of around 7 to 8 due to the bicarbonate ions, And that is all. Thank you very much for listening and have a great day.